And I forgot to hit record again. Okay, here we go. Chapter 16, Control of Gene Expression. So, first off, just imagine this scenario. You're at an orchestra concert, you see strings, you got your woodwinds, your brass, your low brass. Percussion players are waiting for the conductor. The conductor appears, steps up to the podium, and in front of him is the music score, telling which instrument, when it should play, how it should play. Throws up his baton, and the musicians raise their instruments, and all of a sudden the baton comes down, and all of a sudden pandemonium occurs. The tuba starts playing Dixie. You got a violinist who's playing Indigada da Vida. The cellist begins some Mexico national anthem. Snare drums playing Baby Got Back. We got some bass drums, 1812 overture, and the conductor just staggers off the stage, clutching their heart because it's horrible. All right, so got that scenario in your head? Yes. Well, what would happen if in a bacteria cell all of the genes were expressed all at the same time? I mean, obviously that doesn't happen because that would happen. I mean, the bacteria organism just wouldn't even, I would say, probably waste so much energy, it would probably die. So our genes are regulated, they're controlled, they're expressed at certain times in our lives. So you can think of your DNA as a genetic score that expresses different genes at different times. So the objectives for today, we'll talk about regulatory proteins, how it's necessary for the cell survival. We'll talk about transcriptional and post-translational control. So there's two different places where genes can be regulated. We'll again talk about RNA polymerase and how it's linked to the control of transcription, which is going to lead us into an example with the Lac Operon. There are some multiple choice questions on this, so we really need to cover this. We'll describe the two primary kinds of transcription factors in eukaryotes, eukaryote chromosome structure, how it's regulated to gene regulation, and then finally talk about post-translational gene control.
I have a quick question.
Yes.
What happens if we finish the year and we have a week off where we don't have any, when we're done?
We're done.
Really?
Yeah.
Okay. At least it's not like we have to keep going or something.
No. Okay. So section 1 is the control of gene expression. This is kind of like our blueprint for where we're going to go. We're going to talk about how when DNA turns into mRNA, how transcription can be controlled. We'll talk about how when it goes from mRNA to protein, that's translational control. And then after the protein is made, how we can still control the protein by post-translational control. So these are the three kind of broad picture things that we are going to look at, and then obviously dive into nitty-gritty details of each. So control usually happens at that transcription initiation. It's obviously the most logical, but it could happen at any place. We have our RNA polymerase that needs access to the DNA, and it binds to that gene promoter. So we can have things that can block that gene promoter. Okay? There's regulatory proteins that help regulate when genes should be expressed, and sometimes they can block transcription by not allowing the RNA polymerase to bind to the DNA. We also have regulatory proteins that can actually stimulate when the gene should be expressed, so I'd like to call them super promoters. So they can go both ways. So that's one way, using regulatory proteins. When we look at eukaryotes, it's kind of aimed at the process of homeostasis. So remember, homeostasis is maintained in that constant internal environment, where when an organism, it responds to signals around its environment, and then genes can be expressed. So it kind of results in regulating the body as a whole. So a lot of physiological conditions occur here. Other genes expressed in the right cells at the right times in development, like apoptosis. That's why we don't have webbed fingers. Maybe some of us do, I shouldn't say that. But most of us do not have webbed fingers. So
I wish.
Yeah, that's more common.
I wish I had webbed fingers.
All right, 16.2, looking more at regulatory proteins. So proteins, remember, can interact with the DNA, usually through the major groove. And the reason why the major groove is because the major groove is larger than the minor groove. And I have a diagram on the next slide that shows the difference between the major and the minor groove. Remember, our DNA is helix-shaped, and it can unwind for proteins to interact. When DNA is supercondensed, proteins cannot interact with it. So when DNA is unwound, we have proteins that can bind to the outside surface. So sometimes DNA doesn't need to completely unwind. It can still interact with the DNA. Our major groove is the deeper groove. There are nucleotide hydrogen bond donors and acceptors exposed. So
What is the
diagram.
major groove?
What?
What is it?
I have a slide on it. I think it's coming up here, right here. You guys have that?
No.
Oh, I think I had to cut it out because it was too big of a file. Yeah. Okay. So here I have two nucleotides bonded, and I see that I have two two hydrogen bonds. So this is an adenine bonded to a thymine. So this is the major groove. So it's very large compared to the minor groove. And so when our DNA is in a helix, it alternates between major groove and minor groove. So you can see there's more space here for a protein to interact compared to the green line there.
Okay, so it's
Okay,
just on the DNA strand.
Yeah.
Okay.
What physically is that?
It's the two different pyrimidines and purines binding.
Yeah.
I think.
Are you talking about this diagram right here?
Yeah.
Asparagine?
Like I know it's on the inside, but the outside is just like circled in different colors.
Okay, so this is the G and this is the C. So you see these letters here. So I have a G paired up with a C.
Mm-hmm.
And then there is hydrogen bonding going on here, and there's like different other bonds going on. And so this side of the GC is our major groove, and this side is our minor groove. That just means that this side, these two kind of pinch closer together, so the outside opens up more.
Oh. Okay.
Will it always be every other larger, smaller,
Yes.
larger, smaller?
Yep.
Okay, makes sense.
So we have proteins that can squeeze into the major groove. Okay, so when a regulatory protein does that, it has to have certain kind of binding motifs in order to bind with the DNA and interact with it. So we call this DNA binding motifs. We have mentioned the word motifs before,
Repeating sequence or something.
kind of like a theme that reoccurs. Okay. So remember our 4 levels of protein structures: primary, secondary, tertiary, quaternary. When we look at 3D structures of proteins, we notice that proteins that do completely different things have some of the same structures. And some of those same structures are kind of like the alpha loop alpha motif. Okay, so remember motifs, this is just another slide to explain what motifs are. A reoccurring theme. Sometimes we see it in movies or music. So like Christopher Nolan usually has like dark music, and who's the guy? Michael Bay always has like huge explosions in his movies. So like recurrent themes. So analogy here, our amino acids are letters in a language of proteins. Then our motifs would be repeated words or phrases. So like in the movie Star Wars, sorry, I'm a Star Wars geek, this line is said in every single movie. I got a bad feeling about this. That is a recurring phrase in every single Star Wars movie, even the crappy ones. Okay. So some common motifs that are inside of our cells: the helix turn helix, which means it's made up of 2 alpha helical structures. So that's just one common motif, helix turn helix. And then it goes on to describe how this helix turn helix just fits nicely in between the grooves, and it interacts with those sugar phosphate backbone of the DNA, and then kind of gets into the nucleotides a little bit. So here's my alpha helix turn helix shape. And you can see that it fits into the major groove very nicely here. Helix, turn, helix shape. Helix, turn, helix shape. So, other common motifs: the homeodomain motif, which is kind of like the helix, turn, helix, but it's slightly different. The zinc finger motif, where it actually has zinc atoms, and it literally looks like fingers, so it kind of just, I don't know, grasps the DNA. And then we have our leucine zipper model. It's the shape of a Y. The leucine, one common thing is this amino acid leucine is very hydrophobic, so it will not interact with water. So, in summary, there are 4 common motifs that are found in many regulatory proteins. That's just what I want you to understand right there. 4 common motifs. Just know, I think you just need to know the the helix turn helix. The other ones are not important, really. So. Okay. Moving on to how prokaryotes regulate transcription. There are 2 types of control: positive control and negative control. Positive means that it's going to increase the frequency of initiation, so it's going to enhance the binding of RNA polymerase to the promoters. And then negative is the complete opposite. It's going to decrease the frequency of initiation of transcription. I like to think of it as a repressor. It's going to prevent the initiation of transcription. It's going to be a roadblock. So RNA polymerase cannot bind to the DNA.
So the type of repressor is going to change to foreshore.
Mine says repressor.
So where are those at? They're
This is still in the nucleus. Oh, no, this is in the cytoplasm. We're in prokaryotes now. Prokaryotes don't have a nucleus. Okay, so prokaryotes can adjust gene expression in response to environmental conditions because, you know, like they don't have that nucleus, so they're always interacting with their environment, exchanging things, whatnot. Remember that their genes are put together very nicely. They're not spread out like eukaryotes are. Eukaryotes, remember, have introns and exons. Well, prokaryotes don't have that. They code all of their genes together that are all related, so they're all in one spot. So we need to talk about some players in how genes are expressed in prokaryotes, and the first one is called the Lac Operon. So operon just means multiple genes from a single promoter. And in this case, it's going to code for lactose. So one way that the Lac Operon is controlled is through induction and repression. So you see the word repression, which means it's going to block it, and then induction means that it's going to produce more lactose. So bacteria, when it encounters lactose, which is a sugar, a kind of a milk sugar, in its environment, it's going to make enzymes to break down that lactose. So it's going to turn on those genes to break down the lactose it's taken in. It's not always exposed to lactose. It's kind of like a special occasion. But when there's no lactose present, then there's no need to make those enzymes, because why waste energy making those enzymes? With me on that so far? So bacteria have sometimes are exposed to lactose, so they're going to turn on genes to make enzymes, which are proteins.
Is it just lactose that works like that?
No, there's many different types of molecules. But the one that we're just going to focus on, because obviously this is not like grad school, we're just going to focus on lactose. So here's a diagram showing when lactose is absent, the enzymes don't need to be made. So let's just break down what this diagram is showing me. Here I have a DNA strand. Okay. Here I have the structural genes, which is in this blue here, that are going to code for those enzymes that need to be made. Here I have an operator and a promoter. Now the promoter, remember, is the one that's going to promote the RNA polymerase to bind to it. And then we have this operator, which is going to regulate whether or not the gene should be turned on or off. So in this case, when there's no lactose present, this repressor is going to stay on the operator. RNA polymerase, if it tries to bind, it might bind, but it won't be able to move down to the genes. So it's not going to bind. Now, when there's lactose present, notice that this active repressor kind of has these two little loops. Well, the cool thing about active repressors is that they get turned on by the presence of lactose. So when lactose enters the cell, lactose binds to this repressor, and the repressor changes shape, so it falls off the operator. Hey, look, the roadblock has been removed. My promoter promotes the RNA polymerase to bind, then it's going to go past the operator, and then it's going to start reading the genes, making my mRNA strand, which eventually makes my enzymes to break down lactose.
That makes sense.
Isn't that cool?
So,
Like, that just blows my mind. Like, just by one molecule lactose binds to the repressor, and all of a sudden, boom, enzymes are made to break down lactose. So then, those enzymes are going to start breaking down lactose. What's going to happen to the levels of lactose inside of the cell? They're going to go down, and all of a sudden, there's no more lactose. So now the repressor is going to, like those lactose will probably be dissolved or something, and another repressor will come in and bind to that operator and shut off those genes. Yeah, until the next time it comes into contact with lactose. So repression is that the bacteria will not make those enzymes. One type of repressor molecule is tryptophan, which is also an amino acid. When tryptophan, or tryp, is present, the bacteria will not make the enzyme necessary to make tryptophan. So that's an example for repression. Which leads us to negative control. So remember with our lactose, when lactose is present, it prevents the repressor protein from binding to a regulatory sequence. So tryptophan is our regulatory, tryptophan is the repressor molecule in Lac Operon. And I think I have another diagram here. Yes, I do. Okay, so let me just get through this slide and then we'll analyze the diagram. We also have trip repression. So when trip is present, it will cause the repressor protein to bind to that regulatory sequence. If I go back a slide, this diagram doesn't show trip. Okay, so when it is repressed, that means that tryptophan is involved with this repressor. But when there is, if I go back to this slide, when trip is absent, then those enzymes for lactose will be produced. So our Lac Operon is negatively controlled, or goes through negative control. Now our repressor genes tend to bind to the operator. Remember, an operon is a sequence of genes that kind of go together. So in this case with the Lac Operon, these are the genes, LAC-Z, LAC-Y, LAC-A. Yeah. Boom. Okay, moving on to the action of the repressor. Sorry, I don't know why I can't say that word. A protein that can be turned off by operators by binding to the operon, thus blocking the attachment of RNA polymerase to the promoter, preventing transcription. So a repressor just blocks the RNA polymerase from going down to those genes. So nothing new here. Okay, we got to talk about the effector molecule. So we know that our Lac repressor binds to an operator when there is no lactose present. So now our operon is repressed. Shut off. We're not making any genes. An effector molecule is a molecule that binds to a protein and regulates biological activity. In this case, it's lactose. But to be more specific, that lactose is actually called allolactase. So when lactose is inside the cell, allolactase gets produced, and then that's what actually binds to that repressor molecule.
So that's the two little circles that are
The two little circles, yep. And then induction, which means to turn on the genes, begins. And then over time, those levels will decrease, and then our repressor binds to the DNA again. Okay, so that's negative control. Now I'm going to throw one more example at you with prokaryotes, and this deals with glucose. And glucose is regulated by a positive control. So in a glucose repressor, it's going to be repressed when other sugars are present, like lactose. It prefers glucose, but when other sugars are present, glucose will be repressed. So when bacteria is grown in glucose and lactose, glucose is used as the energy source first. Our lactose operon is not induced. When our glucose is gone, lactose is used for energy. So then our lactose operon is induced. So it likes glucose first. So that means the Lac Operon will be turned off. I should say repressed, not induced, because that just kind of messes you up, I think. But when the glucose is gone, then lactose operons will be turned on.
Okay.
There we go. Sorry. Easy like English there, hopefully. Okay. Glucose, when it's produced, glucose is produced when we cleave beta-galactosidase from lactose. Because like lactose, glucose, galactose, they're all kind of related to one another. Because you can like convert from one form to another is what I'm trying to say. Okay. Just to make things even more slightly complicated, I'm going to throw in, yeah, I know, I guess. A major control element is something called a CAP. CAP stands for this word right here, catabolite activator protein. It's located upstream of the Lac promoter on the DNA. So that means here is my promoter. It would actually be kind of like up here a ways.
So how would that stop anything?
What? CAP?
If it's a control on that, wouldn't it stop something?
Well, I was going to get there. What does cap do? I was just talking about what cap was and where it's located. So not all promoters are equal. There are more that are stronger than others. In this case, the promoter that we've been talking about with Lac Operon is very weak. So we have cap that's a very, very strong promoter. How cap makes Lac Operon even stronger, or makes this whole process stronger, is that it binds to the cap binding sites that allows transcription to occur more frequently. So when cap is active, transcription increases. And I think I have a diagram coming up here of how cap does this.
So even though it's a control element, it's like a positive control element instead of like
Yes.
stopping it.
Yep. This is a promoter, so it's going to want to promote the genes. Okay, so here are my operons, my 3 genes, Lac C, Lac Y, Lac A. There's my operator. There's my promoter. And then cap sits right above the promoter. So cap has its own protein called cap protein, which binds to it. And it's going to act as a very, very strong promoter for RNA polymerase to come in, latch on, and then start reading the genes strongly. This happens when lactose is available because we need those enzymes to break down lactose and low glucose. Now, when there's high levels of glucose, we don't need those lactose genes. So then those genes get shut off. So we have that repressor protein. When we have low glucose, which means that glucose is getting transferred into lactose, well, now we have, it starts to kind of open up. We get this C-amp energy molecule, which is getting ready for the binding of RNA polymerase. So low glucose, lactose is still unavailable. It's kind of like this in-between phase. And then all of a sudden, we have high glucose and high lactose. Well, if there's high glucose and high lactose, it's still going to prefer glucose over lactose. So it's very low gene expression. So it's kind of like a faucet with the handle. Is that what you call it, a handle? Because the faucet is where the water comes out, right? So like the knobs.
Yeah, no.
It's kind of like turning it, full blast and then, trickling it. See, this is how you can kind of control it.
So this all goes back to the whole like band thing. Like not all genes can be expressed at the same time.
Right. Yep. So it's kind of like going back to this is actually great. In our orchestra, maybe we just need some, our tuba to kind of lay low a little bit. Like be quiet, just do the nice little beep, 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 beep things in the background or, whatever. It's been a while since I've been in band, so. Okay. So moving on to more in depth, because I just described the diagrams, now these are the words that pertain to the diagram. Cap is regulated by that C-amp. So when you have cap plus C-amp, that means transcription is going to increase. But when you don't have C-amp, then there's no binding that's going to take place, transcription decreases. So even cap has its own regulator, and that's that C-amp. And that C-amp was this nice little box here that you saw. Okay. So another diagram showing C-amp and cap. Here I have my promoter and my RNA. When there's no C-amp, then cap does not bind to the promoter. Transcription occurs at a very low rate. But when they're together, transcription will be very strong. It will occur at a very high rate.
Is there any like higher class than this that like goes into more in depth with all this stuff that we go over?
Oh yeah.
Not here in Wheaton.
But like,
In college, yeah.
oh god, that would be so hard.
Yeah, college biology 12.
I think I'm like struggling to take this in because I don't know what transcription is right now. Like, I'm just like,
Transcription is DNA to RNA.
DNA what?
DNA to RNA. Translation is RNA to protein. Okay.
So let's go into the RNA.
Okay.
when she said translation was on.
Yeah. How about we just skip this slide? Are you guys cool with that?
Yeah.
Okay.
I'm gonna cross it
A
out.
little X in there.
Okay. Let's go back to our trip operon, which I briefly mentioned earlier. So remember, with our trip, it's a repressor. So that means the operon expressed in the absence of trips. So when there's no trip, operon will be expressed, but when there is trip, it will not be expressed. So here we have our operon, our operator with the transcription that takes place with an activator. But trip would be a repressor gene. So TRP would be a negative regulation right here. Okay, moving on to eukaryotic regulation. So that was all prokaryotes. That was all bacteria cells. Now we're going to jump into eukaryote cells, where our DNA is highly organized inside the nucleus and wraps around proteins called histones forming chromatin. So obviously this is going to complicate our protein-DNA interactions. We know that transcription occurs in the nucleus, and there's many different RNA polymerases, 1, 2, and 3, but our RNA polymerase number 2 is the one that's most important here because it binds to the promoter and actually makes the mRNA. So how transcription is regulated is through transcription factors, which I'm going to abbreviate TFs from now on. TFs can be general or very specific. So general means that it's necessary in order for the transcription apparatus to be built and to recruit RNA polymerase to the promoter. So if you take a look at all these like crazy little shapes down here, those are all TFs. If I remember right from like grad school, it takes like 22 different transcription factors to bind to the DNA in order for RNA polymerase to bind. You don't need to know those. But a lot of transcription factors are needed in order to recruit RNA polymerase to the promoter.
So you have lots of transcription factors.
Yes, lots and lots of transcription factors. The transcription factors bind to the TATA box. I'm going to say this again because I'm pretty sure this is a test question.
Oh.
Test question that says something like, the eukaryote promoter is what? And you're going to need to look for the TATA box or the TATAAAT box or something. So
yeah, it's like TATAAAT and then
just look for TATA for the first 4, okay?
TAAT.
So those are general transcription factors. Specific transcription factors increase levels of transcriptions in certain cell types or in response to a signal. So there's maybe specific in different tissues, or they could be time dependent, dependent on the function of that cell. In order for specific transcription factors to help increase transcription, it needs activators to help build that transcription apparatus to recruit RNA polymerase. So it has activators. It also has enhancers that are going to bend the DNA. So here's my promoter. There's my gene that needs to be transcribed. And here's my enhancer. The activators bind to the enhancer, which causes the DNA to bend into this funny little shape right here. And then RNA polymerase binds, and then it starts to transcribe the gene.
So DNA can be in any shape, basically.
Basically. Due to these activators and enhancers. Oh yeah, sorry. Ah. Okay. Ah, okay. And I'll zoom out.
Peasants.
Thank you.
Yep. Sorry.
Boom.
This is a short section right here because tomorrow I'm going to elaborate on eukaryotic chromatin, how DNA actually condenses and uncondenses. It's actually really cool. So we'll cover this in probably tomorrow's lecture. Okay, moving on to post-transcriptional regulation, where we have DNA to RNA to protein. So I'm going to focus in on, after RNA is made, how we can still regulate it before it becomes a protein. So we have small RNAs that can affect gene expression, and these small RNAs are non-codon RNA molecules. So they are not read to make proteins. They are just proteins in themselves, basically. These small RNAs, they are highly structured. They contain several double-stemmed loops, that's their shape, that allows for binding onto certain targets. So I think I might have a picture of this coming up. I hope I do. How small RNAs were actually discovered is that they injected them into C. elegans, and they noticed that certain genes were strongly inhibited. So they were like, wow, these small RNAs actually interfere with RNA turning into proteins. So they kind of interfere with the translational process. So the mechanism for RNA interference or gene silencing, so we have this enzyme called the dicer that generates small RNAs. Basically what it does is it chops double-stranded RNA into tiny little pieces, thus the name dicer. And when this happens, it's creating microRNAs and small interferent RNAs, or siRNAs. Here's where they come into play with regulation. Our miRNAs bind directly to mRNAs and prevent translation. And then these small interferent RNAs are actually the main agents for gene silencing. They actually degrade the mRNA. So remember we said that our mRNA is protected by a cap and a poly-A tail. Well, these siRNAs are going to start trying to degrade the mRNA before it can be transcribed into a translated into a protein. Okay. This diagram right here. Why did I put this diagram on here? Okay, here's my dicer. It's going to make miRNA.
Okay.
Yeah, okay, we're just going to skip this.
Okay.
Moving on. Dicer delivers siRNAs to the enzyme RNA-induced silencing complex, or RISC, which degrades mRNA with the complementary sequence.
Done it now, Max.
I'm gonna have to have a review on what RNA is.
I'm gonna actually have to rewatch every single one of these things because it's just not coming to me.
Okay, actually I'm going to go back to the slide now that I think I know what it's saying. Okay, here's the mRNA pathway, here's the siRNA pathway. So remember, dicer makes siRNA, and the siRNA is going to degrade mRNA. So it's going to degrade it right here. That's what this diagram was saying. Okay, and then we have miRNA, and if we look at what mRNA does, it's going to prevent translations by binding directly to mRNAs. So this miRNA binds to, couples with the mRNA, and it makes it a funny shape. Notice these like nice little hoops here. It's not smooth like this. So the ribosome, once it hits it, it's going to fall off. There we go. Okay, another diagram showing microRNAs, how it's forming these bumps. So when the ribosome comes, it gets blocked. And you can see it here. Here's the mRNA. It's basically going to form all these strands right here, so the ribosome can't. It's a roadblock, basically.
Is this a good thing?
Sometimes. Not always, though.
Basically,
Kind of depends.
like everything that happens in your body.
What was that?
I don't know.
Okay, in one example with humans, we have calcitonin, which controls calcium uptake in bones and teeth. And calcitonin is a hormone. And so it is kind of regulated through multiple proteins from one gene. So if you remember from our spliceosome from our mRNA modifications, spliceosome splices out those introns. Well, when that happens, it sometimes can change the introns and the selected exons that should be removed. Sometimes it removes the wrong thing. So we call this alternative splicing. Examples, real-life examples, calcitonin and CGRP, which is involved in neuroendocrine functions. So here is an example of alternative splicing. Here's my exons. Those are the ones that need to be expressed. And sometimes when it splices it, it puts it in the right order, 1, 2, 3, 4, 5. But sometimes with alternative splicing, it might omit 1, 1, 2, 4, 5, 1, 2, 3, 5. So we call this alternative splicing. Obviously, it's going to change the shape of the protein, as you can see here. We have this like nice little loop, and also we're missing this bottom loop, and this one we're missing the top loop.
So what does it do?
Yeah, it's a defect.
So what exactly happens when what happens to the introns that get cut off?
They get degraded, recycled.
Okay.
So calcitonin, when people lack that certain hormone, they need to take calcium supplements. I actually think Mark has to take calcium supplements.
Yeah.
And then also he has like lactose intolerant.
Yep. Yeah.
Good double whammy.
He has another disease.
So what does that do when you don't get the right stuff? Like you don't get the right loop or whatever in there when it takes
Your protein is altered, so it may not work properly. Maybe it will still take in calcium, but it's not going to work properly, or maybe it won't take up calcium at all, and then you're
So, like,
going to be calcium deficient.
What's a physical example of that certain protein not working? Like, how could it affect you physically? What could a protein do that doesn't, like?
Is it like Mark being like short and really skinny and Sarah?
Yeah. Or like if you're calcium deficient, then let's say you fall down, all of a sudden you break your
Your
easier.
bones can
Yeah.
get really weak.
And then your height will probably shrink over time.
Okay.
So, yeah.
Hardcore.
Herman, if you have to leave, I'll be happy to review this the last 10 minutes, or sorry, the first 10 minutes of tomorrow before Wheaton comes here.
Alright, our bell just rang, so I wrote down what slide we ended on.
Thank you.
So, it's probably like a question you probably should know for sure, but what is the main difference between DNA and RNA? So, like, RNA
Okay.
is half a strand of DNA basically,
Yeah.
and it changes A to U.
Uh-huh.
And it's short term.
And it's short term, because once it leaves the nucleus, it starts to degrade. It starts to get recycled. Everything about the cell gets recycled eventually.
I saw a thing that
Ultimately,
said
what are we studying right
eventually
now?
all.
We are studying how proteins are made. So our DNA is in the nucleus. It never leaves the nucleus. So how in the world are proteins made? So we need an mRNA, which is an intermediate molecule that goes between the nucleus and the cytoplasm.
That's gross.
So once it hits the cytoplasm, that's where our ribosomes are, and ribosomes make proteins. Now this chapter is talking about how those genes are actually regulated. So we've talked about prokaryotes with one example of lactose, how it's positively controlled, how it's also negatively controlled. And now we're talking about how in eukaryotes it's a little bit more complex. So we're going over examples of how genes are regulated. Yes?
Are some of our test questions going to be the ones that we get after class and stuff?
Yes.
Okay, good. At least we know. Study that.
I don't know if like it's just that this chapter isn't as like exciting as some of the other ones, so it's just really hard to take in.
It's
or-
just nitty-gritty details. A lot of kids hate cells. So like this stuff,
Oh, I hate cells.
everyone,
I
everyone,
can't wait for next semester.
I love cells.
Yeah, I'm
This stuff
excited for next
I
semester.
love.
What's next, Lester?
Like
Animals.
and human bodies.
And name mouse.
Yeah.
That's pretty sweet.
Yeah.
So, it's,
that's
so you
how
think
I
it gets
do.
easier? Probably.
Yeah.
This I don't.
Maybe.
I
don't
think so.
take this
I think
at
she
all.
said that earlier in the
It's too complex.
year. But maybe she
Okay.
was lying.
I do this in my mind.
Another way that RNA can be altered, because we've been talking about how it can be altered, spliced in different ways, is that the mRNA itself can be altered, which means that it can change base pairs in the property. Or sorry, base pairs in the mRNA. So instead of an A, it could be a U. Instead of a C, they might change it to a G. Who knows? Remember that our mRNA has to be transported out of the nucleus for translation. And in order for this to happen, it needs to go through that nuclear membrane. It's basically got to have a pass to get through it. So this pass takes place kind of like as a chemical signal or chemical molecule that's going to let it through.
Only 5% reach cytoplasm.
Yeah. Isn't that crazy?
So is that like a 95% just get like
Degraded with inside the nucleus.
So it's just like
Man, we're just a recycling center in here.
it's like D-Day.
If any of those splicing enzymes are attached to the mRNA, it's not going to get exported to the cytoplasm. So this mRNA has to shed all of those enzymes, and then even then, only 5% actually reaches the cytoplasm to be translated into proteins. It's kind of a low efficiency rate. Okay, translation, the start of it can also be controlled. So we have our mRNA in the cytoplasm, and we can still control it there by preventing the ribosome to start translation. So for example, female eggs that are loaded with mRNAs will not be translated until fertilization begins. So when an egg meets a sperm. So it's actually one way. Now remember, our cell is kind of a huge recycling center. Degradation of mRNA is controlled. So the lifespan in mRNA can vary. Sometimes it's like hours to just minutes. So an example of this is casein, which is a major protein in milk produced in the mammillary gland of female mammals. The half-life of it is 1 hour, and little casein is produced. When a female, though, is lactating, the half-life turns into 28.5 hours, leading to that huge increase in that protein called casein.
Alright, I have a question.
So obviously if you are not lactating, casein, little of it is produced, and when it is produced, it degrades very quickly. But when you are lactating, because you have a child, then it has a longer lifespan.
Why is it always half-life? Like, what is the
Yeah,
purpose?
what is
Why
the
doesn't
why?
it say usual life 2 hours? God, I hate that so much.
Yeah,
It
why does it have to be
bothers
fast?
me.
So they like it like that.
I hate it.
Max, anything in this chapter makes sense?
No, that's caspasian or something. It's a different molecule.
Protein from degradation.
It's not feeling any better.
Do I hate how
Okay, so then after the protein is made, we can still regulate it there by degrading it. Maybe we don't want that protein to carry out its functions. So proteins are continually made and degraded, which means that we can alter it chemically to make it non-functional, or we can fold it improperly. Remember, our proteins are very sensitive to its environments, so you can change the pH level, the temperature, whatnot. But the proteins that are old or unused or are incorrectly folded, we need to get rid of those, and that's a typo there, sorry. So we need to get rid of old, unused, incorrectly folded proteins, and how we do that is through proteases. Now, when you guys were extracting your DNA, I gave you some meat tenderizer to sprinkle into your test tube. That was a protease. We were degrading proteins by breaking down those peptide bonds that hold those amino acids together. Now, obviously, these proteases cannot be just floating around freely in the cytoplasm and active all the time, because then
It would destroy you.
your cells would be destroyed. So one way that proteases are contained is through lysosomes. But remember, lysosomes, they're not very specific. They're just general garbage recyclers. So we kind of need a particular protein to know when proteins should be degraded, which leads us to ubiquitin.
I remember that from a while ago or something.
Yeah.
Sure.
No, that was ubiquitin. Then
This is
not
Oh.
ubiquitin.
Yeah, you're right. Yeah.
This is ubiquitin,
Yeah.
not ubiquin.
Oh, Mr. Dealer.
So proteins that have been degraded, or sorry, proteins that need to be degraded are marked with this molecule called ubiquitin. And there's an enzyme called ubiquitin ligase that goes around and it recognizes ubiquitin. So if you have ubiquitin on your surface, that means it's going to use ATP to kind of get to you and break you down. And it's actually really, really cool. So here's my protein. And it needs to be degraded. It's old. We'll just say it's really, really old. So what happens is ubiquitin binds to this protein. Sorry, my mouse disappeared. And then ubiquitin just keeps adding on to the protein, because it's like, okay, this protein really needs to be destroyed. And when it has a lot of ubiquitin, then that ligase is going to find it quicker, basically.
Like a tiger.
Yeah. Okay, so when a series of ubiquitin is added, as you can see here in this blue bubbles here, something called proteasomes come in, and it's a cylindrical shape, and it's kind of like a shredder on the inside. So this protein enters it and just like rips it to pieces inside this cylindrical shape, forming a lot of amino acid sequences as well as peptide fragments. So
Too easy.
pretty
They
cool.
discovered all of this stuff.
Yeah, like
Yeah.
Who in
Well,
God's name had time just to look this up, or just
look it up.
Look
Zach.
it up.
Seriously,
I mean,
I'm
like,
like,
what I mean is like who just had time to
Well,
study
that
all
was
this
probably
stuff.
their life.
They typed it in the magic type. Wait, is there another set of notes?
No, I think I just have pictures. Okay, so in this diagram here, I have ubiquitin, which is this yellow thing, and I guess there's different types of ubiquitin, or it's made up of 76 different amino acids. Anyways, it latches on to here, and I think it's just like step by step. So now it's got ubiquitin, and now it's got more ubiquitin, and it goes into the proteasome, and then it turns into shreds. I don't know what that is. Everything gets recycled. Yep, go get some more. Yep. Okay.
Do we have homework?